Imagine walking into your house, after a really long day.
Oh, I'm already there.
Right. And you reach out, you flip just one single light switch on the wall, and the most structurally impossible thing happens.
Like what?
Well, not only do the overhead lights turn on, but suddenly your fridge perfectly restocks itself.
That'd be nice.
I know, right? And your overall lifespan mathematically increases by a decade, and you vividly recall every detail of a textbook you read 10 years ago.
Okay, wow. Yeah, that defies everything we understand about standard wiring. I mean, one switch is supposed to control one isolated outcome.
Exactly. It sounds like a severe electrical malfunction. Or, magic.
Yeah, I mean, in human engineering, we compartmentalize everything. We build discrete circuits for discrete functions, mostly because, well, it limits cascading failures. If your lights go out, your fridge doesn't explode.
Right, right.
But biological engineering operates under entirely different pressures.
Which brings us back to our favorite microscopic workhorse.
Nah.
So, welcome back to our ongoing series diving into the biology of the nematode C. elegans.
It's good to be back.
And since you are already well acquainted with our transparent millimeter-long star, we are skipping the basic introductions today. We know you know the worm.
We can jump right into the good stuff.
Exactly. But before we get into the incredibly complex cellular wiring of today's study, take a second to hit follow, like, drop a comment, and share this deep dive with your network. It really helps us grow this community of science learners.
Yes, please do.
Because today's mission centers around a brand new study from the journal Science Advances that proves this kind of multitasking master switch, the one we were just joking about, actually exists in biology.
It really is a remarkable paper. It's from a team at TU Dresden, and they've mapped a single neurochemical pathway that acts as this master switch.
And it does a lot, right?
Oh, yeah. It regulates not just sleep, but metabolism, overall lifespan, and even the consolidation of memories.
That's just wild.
It is. And the best part is the molecules they're analyzing aren't some obscure nematode anomaly. They have direct, heavily conserved parallels in human biology.
Right. So we are focusing on a specific neuropeptide called NLP99 and its receptor, NPR16. And, for the sake of making this digestible, you can basically think of NLP99 as the worm equivalent of a major mammalian hormone called somatostatin.
Yeah, and somatostatin is, well, it's a ubiquitous inhibitory hormone in humans. It operates almost like a widespread biological brake pedal.
A brake pedal, okay.
Right. So it regulates the release of growth hormones from the pituitary gland. It modulates insulin release in the pancreas. And it heavily influences our deep, slow-wave sleep cycles.
So it's basically telling the body to chill out across the board.
Exactly. We know it acts as a systemic dampener across multiple organs, but knowing a hormone is important is very different from understanding the precise cell-by-cell mechanics of how it coordinates completely distinct physiological processes.
Right, the actual wiring diagram, which is why we rely on C. elegans. I mean, with exactly 302 neurons, we can track these mechanics down to the individual synapse.
Which is so crucial here.
Yeah. So to understand how this somatostatin equivalent works, we first need to look at how the worm fundamentally toggles between being awake and asleep. And since they don't have eyelids,
right, no eyelids to close.
We have to look directly at the brain circuitry.
And the circuitry is beautifully minimalist. Let's look at the waking state first. This is driven primarily by the AIY interneuron.
Okay, the AIY neurons.
Yeah. When a worm is actively foraging and awake, the AIY neurons are firing continuously. And physiologically, this continuous firing suppresses the command interneurons that are responsible for making the worm reverse direction.
Oh, I see. So the worm moves smoothly forward, just exploring its environment.
Exactly. And as these AIY neurons fire, they are actively secreting our primary molecule, NLP99.
Okay, so AIY is essentially the forward throttle. And as long as it's pressed, it pumps out this somatostatin-like peptide.
Spot on. Now look at the opposing circuit. Sleep in the worm is controlled by a single specialized neuron called RIS.
Just one neuron.
Just one, RIS. For the worm to enter a behavioral sleep state, the RIS neuron has to depolarize. And when it reaches that activation threshold, it dumps a completely different neuropeptide into the system, which is called FLP11.
FLP11. Okay, so I picture FLP11 as like a chemical circuit breaker.
Yeah.
If AIY is the power grid for wakefulness, FLP11 just cuts the main line.
That is actually a highly accurate way to view it. FLP11 binds to specific receptors on the wake-promoting circuits, including those AIY neurons we mentioned, and it opens chloride channels.
Oh, right, so it floods the cells with negative ions.
Exactly. It
Right.
floods the wake neurons with negative ions, hyperpolarizing them so they physically cannot fire. The worm stops moving, stops pumping food, and just enters a sleep state.
Wow, so it's a classic flip-flop switch. Wakefulness, the AIY neurons, actively suppresses sleep. And sleep, the IS neuron, actively suppresses wakefulness.
Yeah, it's a mutually inhibitory loop.
Right, where only one system can dominate at a time, like a two-way radio. But in a biological system, that switch must flip incredibly fast, right?
Oh, it does. And the Dresden researchers demonstrated the speed of this switch using optogenetics.
Which is always so cool.
It is. So by inserting light-sensitive ion channels into these specific neurons, they could force the cells to depolarize simply by shining a specific wavelength of blue light on the worm.
It literally acts as a remote control for the brain.
It really does. So they waited until the worms were in a deep behavioral sleep, and then they flashed the light to manually activate the AIY wake neurons.
And what happened?
Within milliseconds, the worms woke up and began foraging.
Just instant.
Instant. And when they flashed a different light to activate the YI sleep neuron in an active worm, it instantly halted the AIY firing. The mutual inhibition is immediate.
Okay, so the architecture of the switch makes total sense. But the core question of the study is how NLP99, the chemical released during wakefulness, actually dictates the position of that switch.
Right, the actual finger flipping the switch.
Yeah. And this is where the cellular mechanics get kind of complicated, because the effect of this chemical completely changes depending on whether the worm is starving or like sitting in a buffet of bacteria.
Yeah, and this is where it gets fascinating. Survival requires extreme physiological adaptation. So let's examine the starvation context first.
Okay.
When a nematode hatches into an environment completely devoid of food, it enters a developmental diapause called L1 arrest.
Right. It basically pauses all growth to conserve energy.
Exactly. But during this starvation state, the AIY neurons are still secreting NLP99. But the researchers discovered that the receptor for this molecule, NPR16, is being heavily expressed directly on the surface of the IS sleep neuron itself.
Wait, so the wake neuron is sending the chemical signal directly to the sleep neuron?
Yes. And we need to look at the biochemistry of that receptor to understand why. NPR16 is what we call a geocoupled G protein receptor.
Okay, let's break down that mechanism a bit, because geocoupled sounds like dense pharmacology, but I know it defines the entire behavior here.
Right. It essentially defines how a cell interprets a signal. When a receptor is geocoupled, the I stands for inhibitory.
Inhibitory, got it.
So when a molecule like NLP99 binds to the outside of this receptor, it triggers a cascade inside the cell that inhibits an enzyme called adenyl cyclase.
Okay, and what did that do?
It lowers the internal levels of cyclic AMP. And without cyclic AMP, the neuron becomes far less excitable. It struggles to fire.
Ah, okay. Meaning NLP99 binds to the sleep neuron, lowers its internal energy state, and acts as a direct molecular brake.
Exactly.
So it physically prevents the RIS neuron from firing and releasing that FLP11 circuit breaker.
That is the starvation mechanic perfectly summarized. The wake neuron actively suppresses the sleep neuron to keep the worm foraging. I mean, if you are starving, evolutionary pressure dictates you stay awake to find resources.
That makes perfect sense. So
Yeah.
if they took these starving worms and genetically deleted that NLP99 brake pedal, the sleep neuron must have just shut down completely due to the lack of signaling, right?
Actually, the exact opposite.
Wait, really?
Yeah, because it was an inhibitory brake, removing it caused massive overactivity.
Oh, because you removed the brake.
Right. When they deleted the genes for NLP99 or its receptor in these starved worms, the RIS neuron just went unchecked. These mutant worms slept 50% more than a wild-type worm under the exact same starvation conditions.
Wow, 50% more.
Yeah. The wake circuit completely lost its ability to suppress sleep.
A 50% increase in sleep during starvation is a massive behavioral shift. But this introduces the paradox of this study, right? What happens when the environment changes and the worm is well fed?
And this is where biological efficiency becomes just staggering. If you take a well-fed adult worm and knock out that exact same genetic pathway, you probably hypothesize that they would also sleep more.
Right, that would be the logical assumption.
But instead, they sleep significantly less. The sleep states almost disappear.
That is wild. The identical chemical pathway acts as a sleep suppressant in a starving baby worm, but a sleep promoter in a well-fed adult. That suggests the target has moved, right?
That is precisely what the imaging revealed. The researchers mapped the location of the NPR16 receptors in well-fed adults, and they found a profound spatial shift.
A spatial shift.
Yeah. While some receptors remained on the sleep neuron, there was a massive upregulation of these receptors on the surrounding interneurons.
Okay.
And these are neurons that naturally function to inhibit the RIAS sleep neuron.
Oh, wow. So it becomes a biological double negative. It's disinhibition.
Yes. In the well-fed state, NLP99 binds to the geo-coupled receptors on the surrounding neurons, shutting them down. And because these surrounding neurons normally inhibit sleep, shutting them down removes their inhibitory pressure on the RIS neuron.
So by inhibiting the inhibitors, NLP99 indirectly activates the sleep neuron.
Exactly.
That fundamentally shifts how we view genetic function. I mean, the organism doesn't waste metabolic energy evolving a completely new hormone to manage a new environmental context.
Right, why build a new tool?
Yeah, it just shifts the location of the cellular landing pads. It alters the circuit routing while keeping the exact same chemical messenger.
It really is pleiotropy in its most elegant form. One single gene or molecule affecting multiple, seemingly unrelated traits purely based on context.
It's brilliant. But the behavioral switch is only half the story. The time spent in these sleep states directly dictates the organism's physical survival. So we need to look at how the somatostatin circuit controls cellular metabolism and overall lifespan.
Absolutely. The connection between sleep and metabolism is deeply conserved. In mammals, somatostatin is heavily documented as suppressing anabolic processes.
Meaning it stops the body from building things up.
Right. It shifts the body away from building tissue or utilizing stored fat and pushes it toward energy conservation.
Did the researchers find that same metabolic suppression in the worms?
They did. They looked specifically at the starved mutant worms, the ones lacking the NLP99 brake pedal that ended up sleeping 50% more.
Right.
Because they were trapped in an extended sleep state, their metabolic demands just plummeted. The researchers used fluorescent tagging to visualize intestinal lipid droplets.
And just for context, lipid droplets aren't just inert pockets of fat, right? They are highly dynamic organelles. They manage cellular energy flux. They protect the cell from lipotoxicity when fatty acids build up.
Exactly. And in these mutant worms, the lipid droplets were massive. The worms were hoarding almost all their stored energy because the extended sleep states prevented them from burning it on locomotion.
Oh, because they were basically comatose.
Right. And this intense energy conservation resulted in a 15% increase in overall survival rates during the starvation period.
A 15% survival bump just from altering a single neural pathway. That is a massive selective advantage.
Huge.
But what about the well-fed adults? The ones where deleting the gene actually reduced their sleep. Did it harm their lifespan?
Well, the researchers analyzed the lifespan of well-fed adults lacking the NPR16 receptor, and remarkably, their average lifespan actually extended by about 2 full days.
Wait, we have to scale that for people. I mean, a wild type C elegans has an entire lifespan of about 20 days. Adding 2 days is a 10% increase in total life expectancy.
Right. Translated to human demographics, that is the equivalent of adding nearly a decade to average life expectancy.
Well, a whole decade.
Yeah, it is a profound physiological extension. But this raised a really critical mechanistic question for the team.
Which was?
Is the lifespan extension caused by the absence of the NLP99 chemical itself, or is it caused by the altered sleep architecture?
Oh, that's a great question, because if it's just the chemical doing it, you could theoretically develop a drug to block the receptor and just reap the longevity benefits without messing with your sleep cycle at all.
Exactly. So the researchers tested this by creating a double mutant. They took the long-lived worms lacking the NPR16 receptor, and then they additionally deleted a gene called APDEF1.
APDEF1, okay. What does that do?
This specific gene is required for the RIS sleep neuron to function and actually release its FLP11 circuit breaker.
Oh, I see. So they severed the ability to actually fall asleep while keeping the somatostatin pathway broken.
Precisely. And guess what? The lifespan extension completely vanished. The worms died on a normal schedule. So this proves that the NLP99 chemical isn't directly repairing cells or clearing metabolic waste on its own. It is purely the manager. It just flips the switch.
So the actual physiological repair, like the upregulation of stress resistance or the maintenance of proteostasis, that all happens downstream?
Yeah.
Entirely dependent on the organism physically entering that chemically induced sleep state.
You nailed it.
That establishes sleep not as some passive period of inactivity, but as an active, biologically required state for cellular maintenance. If you break the circuit that triggers the state, the body degrades faster.
Exactly. But, the maintenance doesn't stop at lipid metabolism and tissue repair.
Right, there's more.
Yeah, the final piece of this paper looks at cognitive function, specifically the consolidation of memories.
Which is fascinating. The requirement of sleep for memory consolidation is, super well documented in mammalian neurobiology. Somatostatin-expressing interneurons in the human cortex are highly active during non-REM sleep.
Right, and they help generate what we call slow oscillations.
Those are the deeply synchronized brain waves where the neocortex and the hippocampus communicate, right? It's essentially the brain replaying the firing patterns of the day to strengthen specific synaptic connections.
Yeah, it is the physical transfer of volatile short-term information into stable long-term structural changes in the neural network. So the researchers wanted to determine if this highly complex cognitive function exists in the worm and if it relies on the same flip-flop circuit.
Okay, but how do you even measure long-term memory in a microscopic nematode?
Well, you use classical associative conditioning. C. elegans relies heavily on chemotaxis, meaning moving toward or away from specific chemical odors.
Like tracking down food by scent.
Exactly. So the researchers exposed the worms to a specific odor, butanone, and simultaneously provided an abundance of food. Over time, the worm alters its synaptic weights to associate that specific chemical gradient with a nutritional reward.
They learned that the smell of butanone means dinner is served.
Right. Then you remove the food, wait a substantial period, and test if the worm actively migrates toward the butanone odor over other neutral smells. Wild-type worms form this long-term memory really reliably.
Okay.
But when the researchers tested the mutant worms, either missing the NLP99 wake pathway or missing the FLP11 sleep pathway, the memory consolidation completely failed.
Oh, wow. So you need both sides of the circuit intact. I mean, if I'm understanding this correctly, you obviously need the wake state to physically encounter the odor in the food in the first place.
Yeah.
But why does a disruption to the sleep side completely erase the memory?
Because acquiring data and storing data are distinct biological processes. The data strongly suggests that the transition into the FLP11-driven sleep state is the period where the synaptic alterations actually occur. If you cannot activate the AI sleep neuron, you cannot consolidate the associative memory no matter how much you experienced it during wakefulness.
It requires the contrast.
Yes.
The neural network needs the active input of the AI-Y circuit, followed by the systemic silencing of the RIS circuit, to physically restructure the synapses.
Exactly. The delicate, mutually inhibiting flip-flop circuit isn't just about resting muscles. It is a universal, highly conserved requirement for modifying neural architecture.
This single paper completely redefines how we view biological signaling. I mean, we started with NLP99 and its geocoupled receptor, NPR16, and we watched it act as an environmental sensor, altering its target from sleep neurons to interneurons, depending on starvation.
Right.
We saw it manage metabolic lipid storage, driving a 15% increase in survival. We saw it act as the gatekeeper for extending overall lifespan by 10%. And finally, it serves as the necessary toggle required to physically wire new memories into the brain.
It really highlights the extreme efficiency of evolution. A single pathway integrating sensory input, metabolic status, developmental stage, and cognitive function, all to maximize the survival of the organism.
It is incredible how decoding the neural wiring of a millimeter-long worm forces us to rethink our own cellular mechanics. And before we close out this analysis, a quick reminder. If you are a researcher and would like your recent publications explored in a future deep dive, you can contact Dr. Viren Chauhan. We are always looking for literature that pushes the boundaries of how we understand biology.
Absolutely. And, looking at the broader implications of this TU Dresden research, there is a physiological reality we have to confront here.
What's that?
Well, we've established that the exact same neurochemical pathway regulates both the architecture of sleep and the biological aging process. In the worm, chronic disruption of this somatostatin-like signaling prevents cellular repair and shortens lifespan.
Right. Actively accelerates biological degradation.
Exactly. So given that human somatostatin operates through remarkably similar inhibitory pathways to regulate our deep, slow-wave sleep, we have to look at our modern environment. Yeah. We exist in a state of unprecedented artificial wakefulness, midnight blue light exposure, chronic stress hormones, neurostimulants. We are constantly forcing the wake side of our biological switch to remain active.
We are artificially inhibiting our own RIS equivalent.
Yeah.
basically night after night.
We are. And we view sleep deprivation as a temporary cognitive inconvenience, But if this highly conserved pathway functions similarly in humans, we aren't just making ourselves tired.
Right.
We are biochemically overriding the signals required for cellular maintenance. We are actively communicating with our cellular architecture to bypass repair, block memory consolidation, and ultimately shorten our own physiological lifespans.
Wow. That really shifts sleep from a luxury to a non-negotiable biological mandate.
Yeah.
Definitely something to consider tonight when you finally reach out in the dark and flip off that light switch. Until next time, keep digging deeper.